cells, from yeast to those that make up the human body, contain membrane-bound organelles with specialized functions. Mitochondria are double-membraned organelles that harness most of the energy that cells need to grow and reproduce. Nearly all of this energy comes from reactions that take place at the inner mitochondrial membrane. One of the key roles of this membrane is to act as a barrier to positively charged particles called protons, thus allowing a concentration gradient to be maintained where the intermembrane space has far more protons than the matrix. The membrane also contains a large protein complex called the F1-F0 ATP synthase, which uses the proton gradient to drive the synthesis of ATP molecules. These ATP molecules ultimately provide the energy for most of the cell's reactions. Just as man-made power plants produce electrical energy by using the flow of wind, water, or steam to rotate a turbine, the synthase makes ATP by using proton flow from one side of the inner membrane to the other to rotate protein subunits. If there is no proton gradient, synthase subunits stop rotating, and the cell can quickly become starved of energy and die. Therefore, the protein complexes and small molecules that establish this gradient and maintain it play an essential role in the life of the cell. At the heart of this system are 4 protein complexes, numbered 1 through 4. Complexes 1, 3, and 4 directly pump protons from the matrix into the intermembrane space. Complex 2 does not directly pump protons, but it does promote proton pumping in complexes 3 and 4. Proton pumping requires energy, and the 4 protein complexes get this energy by transferring electrons through a series of coupled reactions. This linked process of electron transport is why the 4 complexes are collectively referred to as the electron transport chain. Let's focus on complex 1. A byproduct of sugar metabolism called NADH deposits 2 high-energy electrons in complex 1, where they are passed along a chain of redox centers. Redox centers are clusters of atoms that have different affinities for electrons based on their unique atomic configurations. Let's closely consider a pair of redox centers to reveal 2 reasons why an electron moves from the top redox center to the bottom. First, the bottom redox center has higher affinity than the top one. Second, the distance between these adjacent redox centers is ideal for an electron jump to occur, which explains why electrons typically don't bypass the bottom redox center. A small amount of energy is released each time an electron is passed between redox centers. Complex 1 harnesses this energy across all the redox centers and uses it to pump protons. The last redox center in complex 1 donates 2 electrons to a coenzyme Q molecule. Complex 2 is similar to complex 1 in 2 important ways. First, high-energy electrons also enter complex 2 via a byproduct of sugar metabolism, although here the molecule is FADH2. Second, complex 2 also transfers electrons between several redox centers before donating them to coenzyme Q. One major difference, however, is that complex 2 does not use the energy liberated to pump protons. Coenzyme Q molecules from complexes 1 and 2 donate their electrons to complex 3. One electron is recyclable and can re-enter complex 3 later, but the other passes through 2 redox centers before reaching cytochrome C. Cytochrome C carries the electron to complex 4. The electron transport chain ends in complex 4, where a series of reactions involving 4 electrons converts a molecule of oxygen to 2 molecules of water. The proton gradient is strengthened because 4 protons from the matrix are incorporated into water molecules, and another 4 are pumped into the intermembrane space. In the absence of oxygen, the electron transfer comes to a halt, meaning that ATP synthesis also stops. Indeed, the reason we breathe oxygen is so that it can serve as the final electron acceptor at the end of the electron transport chain. In this animation, we have explored each protein complex in isolation, but in reality they are very densely packed. Together, they effectively make the entire surface of the inner mitochondrial membrane a giant cellular power plant. We are looking at the turning gamma subunit and the epsilon subunits, and there's the C ring in the membrane. The F1 complex is outside of the membrane, while the F0 complex is embedded in the membrane. There's the stalk, including the delta and the B subunit. The alpha subunits are shown in light pink. The beta subunits are magenta. In the middle, the central stalk is rotating. The B subunit stabilizes the hexamer, which means that the hexamer can't rotate. We will now look at a rendering of known structural states and the conformational changes that are happening. The beta subunits are seen as a ribbon diagram. The ADP and inorganic phosphate diffuse into the active site, and the ATP has just been released, and simultaneously the conformational change occurs in the beta subunit. If we zoom into the active site, we can see the molecular structure of the substrates and the chemical reaction. Here is ADP with its 2 phosphates. They are all held in place by a network of hydrogen bonds mediated by the amino acids in the folded structure of the beta subunit. Hydrogen bonds stabilize the base and the sugar, but really primarily the base. There's also a magnesium ion that plays a critical role in not just stabilizing, but also neutralizing the charge of the phosphates. Notice the conformational changes that are happening when the inorganic phosphate comes in. There is the promotion of a planar transition state. Synthesis of a new phosphodiester bond is catalyzed, and then the subunit switches to the open conformation. Upon this reconfiguration in the beta subunit, ATP is ejected from the active site. The enzyme then resets and restarts the synthesis of another ATP molecule. This is a summary of the events at the active site. It is also intriguing just how the C ring rotates. We can start with the analogy of a water wheel. We can easily grasp the idea of how a water wheel rotates. It's driven by the flow of water, allowing the wooden fins to go in a particular direction. Thus, the rotation happens in a specific direction. If the water was to flow in the opposite direction, you would see rotation of the water wheel in the opposite direction. Here we see the C ring and the A subunit. We don't know the full structure of the A subunit, but we know that there's an access channel for protons on the A subunit right in the intermembrane space. and there's a separate egress channel for protons on the matrix side. In this animation, you see protons flowing out from an interface between the C ring and the A subunit. But what drives the specific direction of this rotation? For example, if we were looking down, it is always a counterclockwise rotation. Why and what drives that? On each of the C subunits within the C ring, there is a proton binding site. Notice the high concentration of protons on the intermembrane side and their flow to the lower concentration on the matrix side. Protons load from the highly concentrated intermembrane side onto the C subunits, and they are released on the matrix side where the concentration of the protons is low. But why can't protons go around the other way as well? In principle, just as in the water wheel analogy, the C ring should be able to rotate in either direction, shouldn't it? There are two interconnected reasons for this unidirectional movement. The amino acid side chains in C subunits would repel the bound proton, so that when a proton loads from the intermembrane side, the C ring cannot rotate back in the opposite direction because it is energetically unfavorable. Rotating counterclockwise, on the other hand, is energetically favorable because there are no repulsive interactions. Remember, there's a concentration gradient that favors binding of protons on the intermembrane side and the release of protons on the matrix side. So in summary, the proton gradient determines the direction of the rotation, favoring movement in the counterclockwise direction of the C ring. This avoids unfavorable interactions with the amino acid side chains of the C ring.